Let's talk about membrane transport today. So membrane transport can be categorized in two different ways, passive or active, and then whether or not there's a carrier. So passive or active refers to a gradient. Are the particles moving with or against the gradient? Well, what's a gradient? All right, lots of examples of this all over. Let's look at cows. If you concentrate cows and then you allow them to roam free, they naturally spread out. Okay, they're moving away from each other. So naturally things get over time. That's me paraphrasing the second law of thermodynamics. Everybody knows if you put a drop of food coloring in a beaker of water, those molecules are going to spread out until they reach equilibrium. So describe the gradient of the red particles, assuming that they are penetrating, meaning that they can cross the membrane. So you should be able to look at this and tell me, where would the red particles want to go? Where would they go by themselves? Right? So they're going to move from where they're concentrated to where they are not. Similarly, what about this? Where would the particles go? From where they're concentrated to where they're not. That's passive transport. Active transport is when the particles are moved against the direction they want to go. So can you do this? Yes, we call it active transport, but the particles aren't ever going to do it by themselves. They're going to need help. In other words, they're going to need something to pump or push them. Which brings us to the other way we classify membrane transport. Is it carrier-mediated or not? Carrier-mediated transport, as the name implies, means that the particles can't cross alone. So there's a helper of some sort. We usually draw them like this. So these are channels, pumps, proteins embedded in a cell membrane that are required for the transport. So when you're categorizing these things, you have two decisions. Is there a carrier or not? And are the particles moving with or against their gradient? So if there's no carrier, when particles move in the direction they want to go, that's diffusion and osmosis. We're going to talk about these in a second. There is no non-carrier mediated active transport. You absolutely can't have that. For carrier-mediated transport, we have both. If particles are moving in the direction they want to go, we call it facilitated diffusion. And if particles are moving against the direction they want to go, we call that active transport. And we're going to take a look at all of these next. We're going to start with passive transport. Diffusion, you know, right? These are diffusers. Particles move from areas of high concentration to low until equilibrium is reached. Tons of examples of this. We've got lots of little molecules on one side of the membrane, and as time passes, the particles will equally distribute themselves on both sides. Sometimes particles can't cross by themselves. So here we have cows on two sides of a fence. They need help to do it. They need a facilitator. A facilitator is something that helps. So in this analogy, it would be some kind of a chute that the cows can fit through. Cows are moving in the direction they want to go from where they're concentrated to where they're not, but they need to move through this chute. This is an analogy for facilitated diffusion, and this is how it's normally drawn. So the particles need that protein, but they're moving in the direction they want to go, so no energy is required. Now, osmosis is the movement of water, not particles. The movement of water through a semi-permeable membrane in response to a solute gradient. If you want to see osmosis in your kitchen, grab some gummy bears and soak some of them in salt water and some of them in fresh water and see what happens. What you're watching is the movement of water in response to the solutes inside the gummy bears. Water moves to follow the solute. That's osmosis. So if you set up a situation like this, here the solvent is water, the solute is sodium chloride, and you have a semi-permeable membrane that only allows water and not salt to move through it. Over time, what's going to happen is water will move even against gravity to try and, in a sense, kill that salt gradient. So, flying tip here, diffusion and osmosis are often confused. Don't let them confuse you. Diffusion is about the movement of particles down a concentration gradient. Osmosis is the movement of water from one side of a semi-permeable membrane to another in order to kill a gradient of particles that can't move. So the solvent moves in the case of osmosis. So here's a little diagram to help you out. Here we have solute particles, the red balls, placed in some sort of solvent, the blue balls, and the red balls will move out until equilibrium is reached. They're equally distributed. Osmosis, on the other hand, requires a semi-permeable membrane, and here the solutes, the red balls, cannot cross the membrane. Only the solvent can cross. So you'll get this movement of the solvent through the membrane in order to try to dilute the solute concentration to reach equilibrium. Okay, so diffusion, particles move. Osmosis, water moves. Don't let that trip you up. Active transport. Active transport requires a carrier. That is the protein, but it also requires energy, right? It's not going to happen by itself. The cows aren't going to move from where they are in low concentration to where they are in high concentration. In other words, something has to push them. So again, we require a carrier, that protein, and energy to do this. So this is active carrier-mediated transport. A couple of other specialties, usually with larger particles: endocytosis. Cells can take up relatively large particles by forming an invagination, that's what that's called, and taking substances in and surrounding them with a membrane. We then call it a vacuole. I call this cell gulping. You will also see the term phagocytosis. If you run the same thing the other way, particles that are stored in vesicles can be exocytosed. They're sort of barfed out of the cell through this process. We're going to be seeing this a lot. This is how, for example, neurons communicate, where you see one neuron sending its signal, neurotransmitters, across a synapse to another cell, in this case also a neuron, and the release of those neurotransmitters occurs through exocytosis, cell barfing. As always, I hope that was helpful. Thank you so much for visiting the Penguin Prof channel. Please slam some buttons below. I'll see you in the next one. Good luck.